Hello everyone. We are solving question number 41 from GATE 2015. It says match the drug in group 1 with the mechanism of action in group 2. So we have four drugs in group 1, that is paclitaxel, colchicine, etoposide and methotrexate, and we have several mechanism in group 2. We have to match this drug with these drugs with their respective mode of action. First one is paclitaxel. Paclitaxel is a chemotherapeutic agent. It is used as a chemotherapy in many types of cancer. Paclitaxel accelerates the formation of macrotubule from macrotubule dimer. So what happens? Alpha and beta tubulin make a dimer, and this dimer get organized and form a macrotubule. So it leads to the polymerization of macrotubule. It leads to the formation of a macrotubule by polymerization of these dimer, but it prevents their separation. It prevents the depolymerization of macrotubule, which induces abnormal mechanical reorganization and inhibit normal cell division. So paclitaxel leads to mitotic arrest. This drug not only have an effect on the cell division, but it also inhibit the effect of other factor on the macrotubule system, and together with the stable binding of macrotubule protein, it eventually induces apoptosis. So this drug not only leads to mitotic arrest, not only stop cell division, but also induces cell death. So for paclitaxel, P is 2, that is it inhibits microtubule depolymerization. So colchicine is an alkaloid drug that is derived from plant from lily family. Its mode of action is basically it binds to tubulin and thereby blocking the assembly and polymerization of the macrotubule. So it binds to tubulin. It does not allow tubulin to polymerize and make a microtubule. So colchicine is basically inhibiting macrotubule polymerization. So for Q it is 6. Next one is etoposide. It is a photophyllotoxin derivative. It is used in treating small cell lung cancer. So what it does, it inhibits the DNA synthesis by forming a complex with topoisomerase 2 and DNA. So this complex induces breaks in double-stranded DNA and prevent repair by topoisomerase 2 binding. So there will be no repair. So if there is no repair, the breaks get accumulated in DNA, and if the DNA is not ready, not perfectly, replicated or synthesized in S phase, there will be no the cell cycle will not progress. So cells will not move to mitotic phase. So they will be that lead to halt of cell division and ultimately cell death. So etoposide act primarily in G2 and S phase of the cell cycle. So for etoposide, it inhibits DNA replication. So for R, it is 3. So the last one is methotrexate. Methotrexate is an anti-metabolite. It belongs to the family of anti-neoplastic agent. It is used as a chemotherapy. It is used as a chemotherapeutic agent and as an immunosuppressant in many autoimmune disease. It treats cancer by slowing down the growth of cancer cell. It treats cirrhosis, which is an autoimmune disease, by slowing the growth of skin cells to stop scales from forming, and it also used as a immunosuppressant in arthritis, as it affects the it decreases the activity of immune system. So what it does is it inhibits the enzyme dihydrofolate reductase. This enzyme converts folic acid into tetrahydrofolic acid, which is essential in purine synthesis. So if there is no purine synthesis, there is no DNA replication. So methotrexate inhibit the enzyme dihydrofolate reductase, which is important for DNA synthesis, DNA repair and cellular replication. So methotrexate inhibit dihydrofolate reductase and ultimately inhibit the cell division. So let's match the drugs with their respective mechanism of action. For paclitaxel, it inhibits the microtubule depolymerization, so P2. For colchicine, it inhibits the microtubule polymerization, so Q6. For etiposide, it inhibits DNA replication, so R3. And for methotrexate, it inhibits dehydrofolate reductase. So the answer is B.